Canine parvovirus is a contagious viral disease of dogs. It causes acute gastrointestinal illness, which is most common in puppies between the ages of 6 to 12 weeks. The virus is found in all environments and is extremely hardy. The virus works by weakening the body's immune system and destroying young immune cells, which results in an overall decrease in white blood cell count. This makes it easy for the virus to invade the gastrointestinal tract, where it destroys the epithelium of the small intestine. As a result, the dog will experience severe hemorrhagic diarrhea. Parvovirus is not always fatal. However, if it is a result of dehydration and shock, as well as the septic toxins that are produced by intestinal bacteria.
Possible samples will include fecal samples, blood samples, biopsy of intestinal epithelium.
Preservation and transport of parvovirus samples is a relatively simple process. The parvovirus itself is highly resistant and hardy and does not require buffers, transport media, or specific temperatures to be transported like other viruses or bacteria may need. The samples can simply be collected from site and transported directly to the testing lab. To make a diagnosis, there are numerous tests that can be carried out. Firstly, the history of the patient as well as a physical examination should be carried out in order to take note of the clinical signs that the animal presents. The ELISA, or enzyme-linked immunosorbent assay, is a most common and convenient method for testing in veterinary practices, taking roughly 10 to 15 minutes. Antibodies to the parvovirus are mobilized on the surface of the testing chamber. A fecal sample is then added to the testing chamber and antibodies attached to the parvovirus proteins that may be present in the stool. A color-changing chemical is then added to the chamber, and if parvoviruses are attached to antibodies, the chemical will change color and indicate a positive result. This method is fairly accurate, however does have occasional false positives and means that further testing may be necessary. Another method used to test for parvovirus includes PCR analysis from fecal samples. This test is highly accurate and detects small pieces of viral DNA that are specific to parvovirus in the stool of an infected dog. However, it requires a special lab that performs these tests and much more time is needed. Another method includes a complete blood count. The virus decreases the white blood cells, and thus a white blood cell count that is reduced can be suggestive of a parvovirus infection. Additional tests include serum chemistry, urine analysis, fecal floating, rapid immunochromatographic tests, hematoagglutination tests, and fluorescent antibody tests. Drawbacks to these are that they require special technology, equipment, and time.
Canine parvovirus is a small, non-enveloped or naked virus and has a single strand of negative sense DNA, which is surrounded by a protein capsid of icosahedral symmetry. This means that it does not have a lipid coat covering the 20-faced capsid. Its morphology can only be confirmed through visualization of the virus by the use of electron microscopy. Negative contrast electron micrographs or immunoelectron microscopy are the most commonly used, as they provide the highest image quality. Canine parvovirus replicates well in cell structures of both canine and feline cells, as it is a hardy virus that is not easily destroyed. An example of a commonly used primary cell culture is feline embryo fibroblast culture, or FEF. Growth is most successful when the environment and temperature of the host is mimicked.
Canine parvovirus can be best grown in the laboratory using several primary cell cultures and cell lines such as Madden-Darby canine kidney and Crandall feline kidney cells. The combination of primary cell cultures and cell lines can sustain the replication of canine parvovirus and thereby enable the virus to grow in a laboratory environment. Other viruses than bacteria can also cause diarrhea. To correctly diagnose an animal with canine parvovirus, you need to be able to distinguish it from the other viruses and bacteria. Distinguishing canine parvovirus from bacteria causing diarrhea is relatively easy, as bacteria are much larger than viruses and can be seen under a light microscope. To visualize a virus, an electron microscope is needed. Bacteria have cell walls, whereas viruses only have a protein coat, some viruses even being unenveloped. Another distinguishing factor is that bacteria have a localized effect, while viruses will have a systemic effect. Bacteria can also be treated by antibiotics, but viruses will be unaffected by antibiotics. Canine parvovirus can be distinguished from other viruses by various tests, qualities, and its morphology. It's a single-stranded, negative-sense DNA, non-enveloped virus. The fact that it is unenveloped makes it relatively resistant to disinfectants. In contrast to other diarrhea-causing viruses like canine distemper virus, which are enveloped and sensitive to disinfectants. Another distinguishing factor of the canine parvovirus is that it's stable in the environment, resistant to the effects of heat, detergents, alcohol, and as mentioned above, disinfectants, whereas most other viruses are not. Canine parvovirus DNA can also be isolated and identified by extracting DNA from fecal samples, and it can even identify different strains of parvovirus. Polymerase chain reaction techniques can also be used. It makes multiple copies of a DNA sequence via repeated reactions with a polymerase. This makes it easier to identify the specific virus by looking at its unique DNA sequence. ELISA, or enzyme-linked immunosorbent assay tests, can also be used to detect antibodies specific to canine parvovirus in a fecal sample.
The eclipse phase of the replication of the parvovirus can be explained as the time between the infection of the parvovirus and the appearance of a mature virus within the cell. In the case of the parvovirus, a single-stranded genome must first be converted to double-stranded DNA. From here, a specific mRNA must then be transcribed from the parvovirus viral genome for its successful expression and duplication. Replication takes place in the nucleus and requires host S phase functions. The parvovirus does not encode proteins that stimulate cellular DNA replication.
Canine parvovirus is very prone to mutations as it undergoes continuous and rapid evolution, similar to RNA viruses, in the form of nucleotide substitution. New strain success is dependent on the extension of the range of hosts affected and improved binding to the canine transferrin receptor. Canine parvovirus 1, also known as canine minute virus, was the first strain of canine parvovirus to be discovered. It has minimal pathogenic potential. A few years later, a new strain, canine parvovirus 2, was discovered. This virus is said to be a mutant of another form of parvovirus, possibly originating in feline. There are 3 known point mutations or antigenetic variants of canine parvovirus 2, all differing in one amino acid in the viral capsid protein sequence. Canine parvovirus 2A, this is the first strain of canine parvovirus to replicate readily in feline. Canine parvovirus 2B, this is the strain most commonly found causing the disease and is the most pathogenic strain in some dogs. Canine parvovirus 2C. This strain could possibly be a form of attenuated mutations as it causes mild disease in some cases. In other cases, it is known to cause very severe disease symptoms.
Parvovirus is spread either by direct contact with an infected dog or indirectly by the fecal oral route. Heavy concentrations of the virus are found in an infected dog's stool, so when a healthy dog sniffs an infected dog's stool, it will contract the disease. Even trace amounts of feces from an infected dog may harbor the virus and infect other dogs that come into contact with the infected environment. The virus is readily transmitted from place to place on the hair or feet of dogs or via contaminated cages, shoes, or other objects. The virus can also contaminate kennel surfaces, food and water bowls, collars and leashes, and the hands and clothing of people who handle infected dogs. The virus is resistant to heat, cold, humidity, and drying and can survive in the environment for long periods of time. As one of the symptoms of canine parvovirus is severe diarrhea, the virus is shed back into the environment through the feces of an infected animal.
Antigenic variation. It refers to the mechanism used by an infectious agent to alter its surface proteins in order to evade a host immune response and survive. Latency. Viruses usually mask their presence to the immune system once they have entered cells by directing the synthesis of viral proteins and fragments, of which are displayed on the surface of the infected cell, where they are detected by T lymphocytes. Some viruses enter a state known as latency, in which the virus is not replicated. In the latent state, the virus does not cause disease, but because there are no viral peptides to identify its presence, the virus cannot be eliminated. Resistance to immune effector mechanisms. For instance, some bacteria that are engulfed in the normal way by macrophages have evolved ways of avoiding the breakdown of these phagocytes. They rather use macrophages as their primary host instead of being eliminated. Thank you.